Today's lecture is our third in the series of the adaptive immune response, and specifically allows us to transition to begin discussing in more detail T cells and T cell responses. So just to refresh our memories, to recap what we've discussed with the adaptive immune response, that typically this response occurs within days of an infection, in contrast to what we've seen with the innate immune response that typically occurs within minutes to hours. We know that the response in the adaptive system is highly specific, meaning that there are molecular differences in structure that can in fact be detected, and the specificity allows for detection of pathogens at the species level. We know that the adaptive immune response is highly diverse, meaning that there are a large number of receptors that are involved in the detection at the species level of potential pathogens. We also know that with the adaptive immune response, there is a memory component, which allows for a memory of interaction with a pathogen so that when subsequent infections occur with that exact same pathogen, an immune response can be kicked into high gear almost immediately. It's a much faster response that occurs with the second exposure, as well as much larger in magnitude as far as the response is concerned as well. And we also discussed briefly that the major cell types involved in the adaptive immune response include B cells, T cells, and antigen-presenting cells. And it's those antigen-presenting cells that interact with T cells that we are going to discuss in today's lecture and the following lecture. But before we transition into talking about T cells and T cell responses, I want to add two additional definitions that you may have come across and oftentimes are mentioned when discussing the adaptive immune response. There is what is collectively referred to as humoral immunity. Now, humoral immunity are host defenses that are mediated by antibodies that are present in the plasma, lymph, and tissue fluids. So specifically, when you hear the term humoral immunity, it is in fact referring to the antibodies that are being produced by B cells. To recap what we discussed in the last lecture, we know that naive B cells are B cells that have membrane-associated antibody that have never interacted with an antigen. Once they do in fact interact with an antigen, that then triggers their proliferation to eventually produce memory cells, as well as effector cells. And the effector cells are known specifically as plasma cells, and their job is to produce soluble antibody, that is, antibody that can be secreted from the B cell and can travel through the plasma, the lymph, as well as be found in tissue fluids. It is those soluble antibodies that bind specifically to the antigen that triggered the differentiation in the first place. And we discussed at length in the last lecture, those antibodies have specific functions in the adaptive immune response. They play a role in opsonization, triggering phagocytosis. They activate the complement system, which eventually leads to either phagocytosis or a direct lysis of the cell, the pathogen. They also play a response or a role in neutralizing specific antigens, as well as triggering the ADCC response, which promotes lysis of cells that have been altered in some way. So again, in summary, when you hear the term humoral immunity, it is specifically referring to those B cells, but more specifically those antibodies that are produced by the B cells and what those antibodies are doing in the response. The second term is cellular immunity. And when you hear the term cellular immunity, this is specifically referring to host defenses that are mediated by antigen-specific T cell and various non-specific cells of the immune system. However, in this case, oftentimes when you hear the term cellular immunity, it is almost always referring to T cell responses. We know that there are two broad categories of T cells, which we will review on the next slide. There are helper T cells, whose job it is to secrete cytokines that help play a role in the immune response, and cytotoxic T cells that target host cells that have been altered in some way and target them for destruction overall. So Just like with humoral immunity, your mind should automatically think about antibodies and antibody responses. When you hear the term cellular immunity, you should automatically think about T cells and T cell responses. In one of the first lectures, we talked about the different cell types that are involved in the immune response, and we gave a general definition and the function associated with those cell types. What I want to do is review the T cells in a little more detail because this lecture and the next lecture deals specifically with T cells and T cell responses. So T cells are in fact a subset of leukocytes that are also known as lymphocytes, and they are cells that contain an antigen binding receptor known as a TCR or a T cell receptor. So in this figure, we have 3 different T cells represented on the slide, and each and every one of them do in fact have a T cell receptor. So the TCR is something that is common to all T cells and is going to be the site of interaction with antigen overall. There are helper T cells designated with the letter T and the letter H and a subscript. These helper T cells' job is to secrete cytokines that play a role in perpetuating the immune response. The cytokines that are produced are numerous and have vast functions associated with them, so collectively we typically just refer to cytokines as helping the immune response in some way. So the job of a helper T cell is to produce these cytokines that help the immune response. Cytotoxic T cells are T cells that play a role in eliminating altered or damaged self cells, particularly those that have been infected with a virus or tumor cells. So we've encountered this concept a few times already throughout the semester, the term altered self cell. This is a self cell, again, that has been altered in some way. It could include some type of damage, but in most cases is either growing out of control as part of a tumor or have been infected with a virus. And so those types of self cells do in fact need to be destroyed, and the cells that play a role in that are cytotoxic T cells. These cytotoxic T cells produce molecules known as cytotoxins, and the cytotoxin's job is to destroy that altered self cell. The last category of cells for T cells is referred to as a regulatory T cell, and the job of the regulatory T cell is to help suppress the immune response, specifically repress the immune response against self-antigens. We introduced the concept of tolerance, I believe, in the last lecture, and tolerance is a built-in mechanism to protect the host cells from being recognized as potential threats and having immune responses elicited against them. So the tolerance mechanism prevents the immune system from attacking self-components or self-antigens. The regulatory T cells play a very important role in this particular process of protecting the host components, the self-antigens, from being recognized as a potential threat. In addition, what you see with all of these cells, not only is the T cell receptor present, but there is also a clear additional receptor in these figures present. For helper T cells, that co-receptor is referred to as CD4. That is the same co-receptor found in regulatory T cells. And then in cytotoxic T cells, the co-receptor is a molecule known as CD8. CD4 and CD8 play an important role in allowing for the recognition of antigen to take place, and we will talk about that in more detail in just a little bit. This slide focuses specifically on. So let's recap this concept that I covered in a previous lecture, and that is that T cells, again, cannot directly interact with antigen. They have to have antigen presented to them by a type of cell referred to generically as an antigen-presenting cell. The name implies exactly what the cell does. It presents the antigen to the T cell. In this particular slide, though, what you're seeing is a little more fine-tuning of what is happening in that presentation. So on the far left column, you see labels of molecules and receptors that are playing a role in this process. You have the red dot at the very top of the left-hand column representing the antigenic component, the antigen that is being presented. In this case, it is a peptide, a short amino acid fragment. There are two different classes of MHC molecules, which we will eventually talk about in more detail in our next lecture. So MHC, again, is short for major histocompatibility complex, and these are the molecular components that present antigen to T cells. There are three classes of MHC, but for our purposes, we're going to focus on these two, Class 1 MHC and Class 2 MHC. Class 1 MHC specifically presents antigen to cytotoxic T cells, and Class 2 MHC specifically presents antigen to helper T cells. Going down the left-hand column, you see the T cell receptor, or the TCR. Every single T cell has a receptor, and this is going to be where that recognition of antigen presented to it via MHC and antigen presenting cell is going to take place. And then lastly are those two co-receptors that I mentioned on the previous slide, CD8 and CD4, and these are both playing a role in helping with the recognition of MHC and allowing for this complex interaction to take place. Now, in previous lectures, we've looked at slides that specifically focused on B cell activation, and each time I mentioned that even though the slide focused or had B cell specific components labeled on it, we know that this process occurs for both B cells and T cells. So at this particular point, it's really just my point is to remind you that T cell activation occurs in the very similar way as B cell activation. That is, you have what is known as a naive T cell, and in the slide you can see we have naive helper T cells and naive cytotoxic T cells. The activation occurs when the naive T cell, whether it's cytotoxic or helper T cell, encounters or interacts and detects an antigen for the first time. That activation then allows for the production and the differentiation of the cell types to produce memory cells and effector cells. So there are effector cells for cytotoxic T cells and memory cells that are cytotoxic T cells. There are effector cells that are helper T cells and memory cells that are helper T cells. So both categories differentiate to produce memory components as well as effector cells. So the memory component in both cases, just like we discussed for B cells, are those cell types that are going to play a role in subsequent exposure to this same pathogen. The effector cells, like we said with B cells, are the ones that are doing something. So for helper T cells, the effector cells are the ones that are producing cytokine to help the immune response. The cytotoxic T cells, those effector cells, are known as cytotoxic T lymphocytes, or CTLs, and these are the ones that are playing a role in destruction of altered self cells. With this particular figure, what we are looking at is the T cell activation process in a little more detail. This is a really nice figure because it hones in on the complex interaction that needs to take place between the naive T cell and the cell that is presenting the antigen to it. So on the left-hand side of the figure, you see the naive T cell, and on the right-hand side of the figure is the antigen-presenting cell, the one that is presenting the antigen to the naive T cell. Smack dab in the middle is that complex association that we've been talking about repeatedly now, where you have the antigen, as represented by the red dot, being presented by MHC, which is part of the antigen-presenting cell, and that complex interaction has to be received by the T cell receptor. In addition, you see the co-receptor that is playing a role in recognition of MHC, and so this very first step is the first signal that occurs to allow for activation of the T cell to take place. This is antigen-specific TCR engagement that occurs as step 1 or signal 1. The second signal that is also received to help with the activation process is contact with co-stimulatory receptors. There are additional receptors on both the naive T cell as well as the antigen-presenting cell that play a role that interact with one another to produce a second signal, which is co-stimulatory, again, allowing for this activation process to take place. Lastly, the third signal is signal from cytokines that allow for differentiation to actually occur. Now let's look at a little more detail associated with helper T cells. So it turns out that what we typically do in this course, and that I like to focus on, is to collectively lump the helper T cells together as one category of cell that plays a role in producing cytokines. However, as you can imagine, it is much more complicated and complex than that. There are several different categories of helper T cells, and they are all listed on this slide for you. Now, I won't ask you to memorize specific helper T cell types or names and what their functions are, which cytokines they produce, but I like to highlight this again to remind you that it is much more complicated, and that hopefully what we're doing is simplifying to get a good general understanding of what's happening. But what you can see here is that you have what is referred to as the naive helper T cell that will eventually differentiate to produce the cells that you see here. The naive helper T cell, before activation, can also become a regulatory T cell, and we will look at that fine-tuning a few lectures down the road when we start to talk about immune dysfunction, as well as what the role of regulatory T cells are. More importantly, for this particular slide, all of the other cells that you see here, again, are distinct classes of helper T cells, and they do in fact play different roles overall in the immune response. If you look down at the very bottom of the slide, you can see the effector functions in the boxes, as well as the role in disease overall. So for example, effector functions of the class 17 helper T cells, these combat extracellular pathogens, bacteria, fungi in barrier tissues. The next one looks or combats helminth infections, which are worms, and activates eosinophils. And again, you can see there are some specialties associated with each of the different classes of helper T cells. But again, for our purposes, we will lump helper T cells together as a group or a family of cells that produce cytokines to help the immune response. So all T cells have a TCR, which is short for T cell receptor. The T cell receptor is the structure that allows for receiving the signal of the antigen presentation that occurs to the respective T cell. What you see on this particular slide is a comparison between the B cell receptor, which is known as the antibody, and that's on the left-hand side, and on the right-hand side is a typical T cell receptor. My point here is to really just highlight some differences between these two, but also highlight some very important similarities between the two. The obvious difference is that the B cell receptor, the antibody, is a much larger molecular structure in comparison to the T cell receptor. What I want to highlight, though, are the similarities in that both components or both structures have variable components as well as constant. So when we discussed antibodies, we said that those constant regions vary little or change very little in their amino acid composition. Hence, they're referred to as being the constant portion. And the V portion is the variable portion. This is where a lot of differences occur, and this is where the specific binding of epitope of the antigen takes place. So those tips of the Y in the antibodies is where the variable regions are, and that is specifically where that interaction with antigen occurs. This is the same as what we see in a T cell receptor. The very tip of the structure has the variable portion, and this is the portion that will in fact interact with antigen. Both structures have a transmembrane domain, which allow for the association with the respective membrane, either the B cell or the T cell overall. So now we'll transition just a little bit into the nomenclature associated with the cells that are doing the presentation of antigen to the respective T cells. So not listed on this slide is the first concept that I want to get across, and that is cells that display antigens with class 1 MHC to cytotoxic T cells are referred to as target cells. So up until this point, we have been calling any cell that presents antigen to a T cell an antigen presenting cell. But now we can fine-tune that terminology in that the cells that present antigen to cytotoxic T cells are referred to as target cells. So even though their job is to present antigen, the specific nomenclature associated with those cells is that they are referred to as target cells. So let me say that again. The cells that present antigen to cytotoxic T cells are called target cells. Then if we transition into the cell types that display or present antigen to helper T cells, those are referred to still as antigen presenting cells. Now, the antigen presenting cells can come in two different categories. There are those that are referred to as professional antigen presenting cells, and those are just cell types that their number one job is to present antigen to your helper T cell. In the professional antigen presenting cells, the molecular structures that are allowing for receiving the signal as well as sending the signal are always going to be expressed. So this is their number one job overall, is to present antigen. And you have some examples of that listed here on the table. In addition, there are antigen presenting cells that are considered non-professional antigen presenting cells. And the easiest way I use to describe this is that these are cells that have some other normal function other than playing a role in the immune response. So some of the examples you see up here are fibroblasts. These are cells that are part of the skin. Glial cells, cells that are part of the brain. Those cells' normal job is to either be part of the skin or part of the brain, like some of the other examples you see here. However, these cells can also present antigen to helper T cells. And so these are again referred to as non-professional because their number one job is not to be playing a role in the immune response. It's something else, but they can also have sort of a side gig where they can also present antigen to helper T cells. So now we'll transition a little bit into talking about the major histocompatibility complex, or MHC molecule. Like I mentioned in the previous slide, there are in fact three classes of MHC: class 1, class 2, and class 3. Class 1 is expressed by nearly all nucleated vertebrate cells, and the job of the class 1 MHC is to present antigen to cytotoxic T cells. So based off of this, plus the terminology we just talked about, those cells that are presenting antigen to cytotoxic T cells utilizing a class 1 MHC are going to be called target cells. Class 2 is expressed only by antigen presenting cells, and this group of cells presents antigen to helper T cells. The last class is class 3, and that is distinct from class 1 and class 2. This plays a role in complement system activation and also allowing for some cytokine production. However, we're not going to focus on class 3 for this particular lecture or this particular class. What we really want to do is focus on the first two, class 1 and class 2.